According to the U.S. National Library of Medicine, X-linked SCID is the most common form of SCID. It affects 1 in 50,000 to 100,000 infants each year, causing them to be born without an immune system. Now, because they were born without an immune system, these infants are much more prone to life-threatening illnesses and diseases.
There is currently a study underway in which bone marrow stem cells from X-linked SCID were taken from patients and inserted with the correct gene sequence via retrovirus. The cells were then injected back into the bone marrow in hopes that the new cells would produce functional T and B lymphocytes. The study has not shown results yet. However, there are other studies whose results show that gene therapy is a promising cure for those with X-linked SCID. In a recent study, 9 boys with X-linked SCID underwent a gene therapy using a vector engineered to cure the disease and prevent leukemia from resulting from the therapy. 7 of the boys have acquired a functioning immune system without developing cancer. They are still being monitored, however. Neither of the other 2 developed cancer, but the gene therapy was ineffective.
The current go-to treatment for X-SCID is a bone marrow transplant during infancy. This procedure is painful and it has varying rates of success. If the bone marrow is not well established in the bones of the patient, then lymphocyte production will be too low and lead to a normal life. In our treatment, all lymphocyte-producing cells will be exposed, increasing the likelihood that multiple colonies of healthy cells will form and begin producing fully functioning white blood cells. The main improvements to this method will include less pain, less invasiveness, earlier administration, and the potential to have multiple bones producing healthy T and B lymphocytes. This would lead to higher potentials for the immune system production, and in some cases may even allow the patient to lead a normal life with a completely functioning immune system.
Okay, the mutation in our gene is caused by multiple different mutations in the IL2RG gene, which codes for a protein on the membrane of the T and B cells. And there's a lot of different possibilities that can happen with the gene sequence, but the one that we focused on with this project is a point mutation right here at the start codon. It changed from an A to a G, and a deletion here with these T and G, which causes a frame shift mutation. So the reading frame is messed up in the gene, causing the protein to become defective.
The areas of the body targeted would include bone marrow, the spleen, and lymph nodes. These locations are where T and B lymphocytes are created. More specifically, the stem cells in these tissues would be targeted, as these are what divide and mature into lymphocytes of interest. Because testing has never been conducted on strictly gamete cells, only somatic cells will be targeted for this test. The method of delivery would be specifically targeting retrovirus, which has been used in other gene therapy trials and shows promise for gene delivery. Typically, viral vectors are confronted by the human immune system, but in SCID patients there will be little or no immune system to fight the viruses from reaching their target cell and administering the genes. Once the retrovirus injects its RNA, the cell should be able to produce DNA based off of the RNA code. This will give each of the affected cells a correct copy of the DNA, and thus they have become functional. In order to keep the treatment functioning over a period of years, a strong promoter will be included in the RNA sequence, which complies with the fact that the promoter is already mutated and needs to be treated. This version of delivery was decided upon because it is one of the most efficient in producing and in delivering the genetic material into the targeted cells. Also, viruses will not be confronted in SCID patients, theoretically allowing for a higher efficiency. A retrovirus was chosen in that it creates a DNA copy of the dividing cell, which will allow for a longer treatment. The virus would need to be altered in a lab to remove all pathogenic effects and insert the desired genetic materials. It has not been determined if this will be achieved through biological manipulation or another method such as microrobotics. In either case, the virus will need to contain the normal gene sequence and the promoter region.
Upon the reception of the viral RNA, the cell will begin to make a DNA copy. The DNA copy will have a promoter attached, so DNA synthesis will occur. The normal proteins for white blood cells will be created, whereas before the protein was a messy string of amino acids and codons that couldn't work because of all the mutations associated with X-SCID. The correct proteins should be present in all lymphocyte offspring of treated cells. After administration, blood tests could be run to measure levels of the protein in blood cells. With normal levels, the immune system would be functional. The corrected protein would allow the lymphocytes to communicate as normal and thus work together to ward off pathogens. The patient should no longer be prone to opportunistic, weaker pathogens.
There is a barrage of technical hurdles to face in developing such a radical approach to treating SCID. Tests in mice have shown that the genes themselves may cause tumor growth or cancer when used in gene therapy. There have been many studies showing that leukemia can be a result of X-SCID gene therapy. The exact sequence of treatment cannot be the same for every patient, as each mutation is unique and must be dealt with. This would increase the time, cost, and chance of error when actually delivering the treatment to the public. The treatment would be delivered by a virus, meaning there are several risks. Mutations within the virus would cause it to become ineffective or become a pathogen. In a patient with SCID, it would most likely lead to death. The actual virus would also be hard to engineer because of the degree of stability to where it would be considered safe for the public. Ethics would also be challenged, as this version of the treatment would be targeting an unborn fetus. The controversy over abortion and super babies would be both pulled over into this treatment, meaning it may not be accepted by the public and also may face challenges becoming legal.